Hello, everyone, and welcome to this session of the Atheronet Journal Club. Today, we have the pleasure to have with us some great researchers from collaborations within Atheronet, and I'll let Christina to do the honors to introduce our speakers today. Over to you.
Here I am. Can you hear me?
Uh-huh.
Okay, perfect. So it's our great pleasure today to have Eva Jover and Andrea Caporali, who just recently published their work in cardiovascular research on the loss of microRNA 26B promotes aortic calcification through the regulation of cell-specific target genes. So we're super looking forward to your talk. And without taking too much time, Eva, please go ahead and start.
Okay, thank you. So before starting, I would like to thank all the people organizing this Atheronet Journal Club meetings and for inviting us to discuss the paper that we recently published in cardiovascular research. I will be presenting this paper entitled "The Loss of microRNA 26B promotes aortic calcification through the regulation of cell-specific target genes" on behalf of all the people contributing to it, and from Scotland, USA, and Spain, in the case of my former PI, Natalia Lopez Andres, and myself. Also, I'd like to add that this was the PhD project of the main author, Diana Luna Buitrago, and that it was led by Andrea Caporali. And also, I'd like to say that our collaboration started like in 2022 when I joined Caporali's lab for a postdoc as a postdoc visitor for 2 months to do some histological analysis on the microRNA 26B knockout mice model. Over there. And I have to say that personally and professionally speaking, it was very rewarding. And since then, we have been collaborating to test the roles of this microRNA in different cardiovascular contexts. These are my disclosures. And before getting started on the science in this manuscript, I would like to give some few comments on the disease and the mechanisms involved in vascular calcification. First of all, vascular calcification consists on the accumulation of calcium and phosphate in soft tissues. It can happen, it can affect large and medium-sized arteries, but it also can happen in the heart valves. And while the prevalence is not really clear, a recent publication was establishing that it may affect 24% of middle-aged general population. Different factors like the age or the biological sex might be acting as risk factors for the development of vascular calcification. For example, it has been reported that there is a 90% prevalence of coronary artery calcification in males and around 70% in females when they are in their 70s. But other clinical conditions such as atherosclerosis, diabetes type 2, chronic kidney disease, hyperlipidemia, or hypertension might be also underlying the development of vascular calcification. It's characterized by long preclinical symptomatic stages, which later in life may lead to the onset of clinical symptoms associated with a poor prognosis and severity risk. And the severity, both the symptoms and the risk associated with this vascular calcification might be really depending on both the location of the calcification and the type of or the nature of the calcification in terms of micro or macro calcifications. Either way, no matter the clinical outcomes that we are getting from vessel stenosis or vessel stiffening, vascular calcification has been associated with an increase of cardiovascular mortality and all-cause mortality, no matter which is the vascular bed affected. And basically, in the physiopathologically context, what we have is a vascular to bone tissue replacement, as it has been demonstrated by the overlapping in Raman spectrometry, comparing bone and calcified vessels. And this is mostly happening because synthetic phenotypes in the vascular smooth muscle cell become persistent because of the continued vascular injury exerted by aging or the clinical conditions we were mentioning before. And in the absence of inhibitors like pyrophosphate, MGP, or phetuin A, these synthetic smooth muscle cells may undergo two main clinical hallmarks of the vascular calcification, which are osteoblast-like differentiation and an extensive matrix remodeling, including calcification. So we know all these, and we know that these factors are going up and those are going and other factors are going down, but how they are happening, we don't know yet. So in this context, microRNAs are emerging as relevant factors, being able to regulate different multiple targets belonging to the same signaling pathway in the context of calcification by inducing or reducing the osteoblast differentiation of the smooth muscle cell. Moreover, we previously identified the protective role of microRNA 26B in the vasculature with a downregulation associated with a pathological condition such as ischemia injury, and with the rescuing of the microRNA 26B expression associated with an improvement of the endothelial cell function. This in vitro, in vivo was also demonstrated to be associated with a protection of the protection of.
In the past 20-25 years, we had an increase.
Sorry, Paolo. Could you
let
let me mute him? Then.
Yeah.
Sorry. You can
Okay.
go ahead.
Yeah,
Thank you.
I go on. Okay, so this microRNA 26B rescuing was also associated with a protective effect on endothelial and myocytes in the ischemia by reducing the apoptosis rate related to ischemia. And additional bioinformatic prediction analysis identified that BMP and TGF-beta signaling pathway might be significantly or potentially regulated by microRNA 26B. With all that in mind, we aim to investigate whether and how microRNA 26B cell-specific target genes and vascular cell-to-cell communication could initiate and promote vascular calcification. So we collected human aortas from patients with aortic valve disease and aortopathy requiring concomitant valve replacement and aorta replacement, as well as patients with lone aortopathy requiring a valve-sparing, aortic root replacement. And the samples were classified, the tissues were subgrouped as low and medium or high calcification levels by the digital image analysis of histological preparation stained with alizarin red. And also we performed some ASEAN blue and Sirius red stainings for identifying that there is a decrease in the collagen content and an increase in the glycosaminoglycan expression in medium and high calcified samples. We also looked at the expression of microRNA 26B, which was reduced in medium and high calcified samples, and we actually found a significant and negative relationship between microRNA 26B and the degree of calcification quantified by alizarin red digital image analysis. So to characterize which was the phenotype, the cardiovascular phenotype of microRNA 26B, we developed a knockout mice model using CRISPR-Cas9 technology that was creating a partial relation of the seed sequence of the microRNA 26B. And in these samples and in these animals, we collected aortas from 3 and 6 month old wild type and knockout animals, and these were probed with 18F and NAF probe before being imaged by PET CT scan. Only after 6 months, we found an increase in significant increase in the microcalcification presence in the aortic arc and thoracic aortas, and that was further validated using different methods such as histological preparation analysis with alizarin red staining or calcium deposition quantification using ochrosephthalane staining. Even though we found the presence of microcalcification in 6-month-old mice, which may suggest that microRNA 26B knockout spontaneous calcification is associated with age, we didn't find a different expression or patterns of elastic fiber density or fragmentation in histological preparations stained with elastica vangiosum. Moreover, we analyzed the deposition of the fibrosis content using Sirius red staining. And as you can see here in these bright field images and these two graphs here, the knockout of microRNA 26B was associated with an increased thickness of the intima media, but also with an increase of the medial fibrosis. And also that was associated with the expression of thicker fibers as seen by these polarized light images. So all that may lead to the development of aortic stiffness. And to further characterize that in a functional level, we performed some pressure myography, which in this case was showing a decrease in the strain of aortas in knockout mice, suggesting a loss of elasticity, but also it was tested by atomic force microscopy, and we found that it was associated with an increase in the Young's module, suggesting that it actually was associated with an increase in the aortic stiffness. So we then performed single-cell transcriptomic analysis on cells isolated from 6-month-old wild-type and knockout mice to map the cell contribution to these different phenotypes. And we identified 12 clusters that were assigned to their putative identities, and also the hierarchical expression of the different expressed gene signatures that we found between knockout and wild-type mice. The knockout of the microRNA was associated with significant changes in total cell distribution and also the cluster frequencies. And then to identify if there were microRNA 26B enriched target genes pathways that could be explaining these different phenotypes in between the wild type and the knockout mice. We performed some additional network analysis integrating the upregulated genes found as differentially expressed and predicted target scans targets. And as you can see here, for smooth muscle cells, endothelial cells, and fibroblasts, the key nodes for smooth muscle cell was SMAD1, associated with osteoblast differentiation and BMP signaling. For endothelial cell, it was collagen 1A2, associated with extracellular matrix organization. And for fibroblast, it was GATA4, which was associated with regulation of gene expression. So to understand whether this regulation of SMAD1 was actually happening in the smooth muscle cell, and it was specific for smooth muscle cell in knockout mice, we performed some additional expression analysis. And as you can see here, only the downregulation of microRNA 26B in smooth muscle cells was associated with a significant upregulation of SMAD1. That was confirmed as well by immunocytochemistry. Here you can see how how was active the nuclear translocation of phosphorylated SMAD1 in the vascular smooth muscle cells, but not in the intima. And it was also confirmed by semi-quantitative Western blot analysis looking at phosphorylated and non-phosphorylated SMAD1. And because SMAD1 is a downstream effector of BMP ligand to BMP receptor signaling, we did some additional analysis looking at trying to test if there must be a cell-to-cell communication, confirming that this SMAD1 is activated in the SMAD muscle cell and in the knockout mice. And as you can see in this cord diagram, cluster 9, that was corresponding to fibroblast, was really strongly signaling through to cluster 8 and cluster 0, which were associated with SMAD muscle cells, and also somehow to the endothelial cells in cluster 6. And more specific analysis indeed revealed that fibroblasts might be acting as senders and influencers, while SMAD muscle cells might be the receivers and influencers associated with the specific BMP phenotypes that we described for the knockout mice. Moreover, when mapping the BMP pathway, the BMP ligand and receptors across the different vascular cells, we found that overall BMP4 must be upregulated in fibroblasts, also some other ligands like BMP2 in here, but mostly BMP4. So we conducted some additional experiments in fibroblasts, smooth muscle cells, and endothelial cells sorted from wild type and knockout aortas. And as it was expected, BMP2 was the most upregulated ligand of the BMP signaling pathway in the fibroblast. So in order to understand if this BMP4 released by fibroblasts could be the one inducing the expression, the downstream expression, not the downstream expression, sorry, the downstream signaling of SMAD1 in vascular SMAD muscle cells, we conducted some additional in vitro experiments in which we used conditioned media from fibroblasts isolated from knockout aortas. And well, as you can see here, the conditioned media from knockout fibroblasts were associated with an upregulation or an increased pSMAD signaling seen by immunocytochemistry in SMAD muscle cells, while using an inhibitor, LDN 193189 inhibitor from now on, LDN, that is inhibiting specifically ALK receptor signaling, was associated with a decrease of SMAD1 nuclear translocation in the SMAD muscle cell. These findings were also corroborated by Western blot semi-quantitative analysis in here. And importantly, it was associated with the expression of these two markers, MFAP4 and ID3, which were associated with the signaling of BMP4 in the SMAD muscle cells. I haven't showed this data because it was a bit redundant with this figure, but I'll explain you now. So basically what was happening is that because of the content of BMP4 in the conditioned media from fibroblasts isolated from knockout aortas, from mice with from knockout microRNA-26B aortas, these two targets were upregulated as downstream effectors of the BMP4 signaling, while blocking the signaling through the ALK receptors, and so blocking the signaling of BMP4 was associated with a decrease. And importantly, for the clinical outcome, in the figure D and E, you can see that this inhibition of ALK receptor signaling was associated with a significant decrease in calcification when the conditioned media from knockout fibroblast was supplemented with this LDN inhibitor. And this happened in two different contexts, using both fibroblast conditioned media, but also in a co-culture system in which we used transwell plates. And the bottom plate was seeded the SMAD muscle cells, and then on the top one was seeded the fibroblast. So we demonstrated this anti-calcific effect of LDN into different crosstalk, cell-to-crosstalk models, let's say. So after seeing this effect and this positive and beneficial effect against the development of calcification, we performed some additional in vivo experiments using the inhibition of ALK receptors in our microRNA 26 knockout model. And as you can see here, addition of the LDN was associated with a decrease here and here, a decrease of aortic arc calcification and thoracic aorta calcification. And that was further confirmed by looking at the calcium deposition measured as ochrosophthalane, using the ochrosophthalane method. And importantly, this effect, this LDN effect, was associated with a reduction of phosphorylated SMAD1 nuclear translocation in SMAD muscle cells, as demonstrated by immunocytochemistry here and image quantification in this plotted graph here. So this relationship between microRNA 26B and SMAD1 was also demonstrated in two different cohorts of human samples. First, we found higher expression of SMAD1 associated with the higher expression of calcium in high and medium calcified samples, and an inverse negative, and significant relationship with the expression of microRNA 26B. These samples were the previous ones I showed in my first slide. And also we found that this relationship between the 26B and SMAD1 was found in an additional cohort of patients with aortic aneurysm, in which microRNA 26B was calculated using in situ hybridization here. And SMAD1 was found negatively associated with the expression of microRNA 26B in low or moderate calcification samples, in this case quantified by Boncosa staining. And here you can see in this graph that it was actually as higher was the expression of SMAD1, smaller was the expression of 26B in low, and the other way around in medium calcified samples. So this is the graphical abstract summarizing the data that we collected in this manuscript. We developed a microRNA 26B knockout mouse model, which was studied for the development of spontaneous microcalcification using PET-CT scan. We performed some bulk RNA and single-cell RNA sequencing to find the contribution of different cell types and the possibility of crosstalk between the vascular cell types. We found that SMAD1 was at the center of the network analysis associated with SMAD muscle cell calcification, and that it was a result of BMP4 released from fibroblasts without not expressing microRNA 26B, and that translated in the development of moderate and high calcification in human samples in two different cohorts, which gives strength to our findings. And so this manuscript was submitted to cardiovascular research, and we got a few comments that I particularly found very interesting, because they were addressing some previous findings in other contexts of the regulation of microRNA 26B or the pathology itself. So one of the questions was to perform sex-dependent analysis of the phenotype in microRNA 26B knockout mice. And as we show in here, there were no significant differences in terms of the development of microcalcification in 6-month-old females, as seen by the significant upregulation of microcalcification in aortic arc or thoracic aorta. So there were not significant differences to the male knockout counterparts. Regarding this, we were also asked to improve the patient's table. At the beginning, we were just showing the total statistical information regarding age, aortic stenosis, diuretic, drug medicine consumption, etc. And basically they were asking us to specify the number of male and female subjects, and in each group of low or medium high calcification subgroups that we identify by digital image analysis of alizarin red histological preparations. We didn't find significant differences in this context, so it goes in agreement with our data in the animal model. We were also asked to find if there was a correlation between the expression of microRNA-26B and the lipid profile in the patients studied. We didn't find any association. This data was only provided to the referees, and actually the question was quite pertinent because microRNA-26B has been associated with the lipid profile in some severe forms of hepatic lipid disease, fat disease, sorry. So, and we were not asked for this question, but because statins can affect the content of can affect the lipid metabolism, we also did some additional analysis, and we studied the expression of the microRNA-26B in patients having or not statins, statin regime. And again, we didn't find a relationship with the lipid profile, even though the total cholesterol levels and the LDL levels were reduced by the statin use. We were also asked to include volcano plots to visualize the upregulated and unregulated genes, and for that we generated MA plots to visually represent the differentially expressed genes. Here you can see, and it was done across the different vascular cell types. And in red color, you're having the increased genes, and the decreased genes were shown in green color. And we also provided the potential biological implications associated with smooth muscle cells, endothelial cells, and fibroblasts, with a major contribution of or regulation of transcription by RNA polymerase II in smooth muscle cells, or cell migration in endothelial cells, and cell junction assembly in fibroblasts. We were asked whether human microRNA-26B level was associated with the aortic fibrosis, as we saw for the in vivo samples. And while collagen content was negatively and significantly correlated to the calcification degree, and glycosaminoglycans were positively associated with the calcification degree, we did not find an association with the levels of microRNA-26B, and that might be resulting from the different stages in which we are analyzing calcification. We found that microRNA-26B must be more related to the early development of calcification, while the aortic samples that we are working with, they are very advanced. So probably we are having an inconsistency because of the different stages that we are looking at in these human and in vivo samples. And finally, we were asked which was the specificity of LDN, because as I was saying, is an ALK inhibitor, ALK receptor inhibitor. And so the question was answered as shown in here. LDN is targeting LKA2 or ACVR1, and ALK3 or BMPR1A, so it's targeting bone morphogenetic protein receptor type 1. And it's cell permeable, which will help to our model as the integrity of the endothelial cell layer was intact. It inhibits specifically the BMP receptor-dependent phosphorylation of SMAD1, 5, and 8, which was the one activated in smooth muscle cells from knockout mice. And also, if we go back to one of the figures I was showing with the mapping of the BMP pathway, depending on the cell type, we found actually that ACVR1 was significantly upregulated, and as well BMPR1A in here was upregulated in smooth muscle cells. So it could be mediating the effect of BMP4 released from the knockout fibroblasts. Other questions that we were asked were to increase the number of biological replicates to 5 for the specified figure panels. This was mostly associated with the statistical robust analysis. We were asked to provide Western blot for quantification related to the expression of phosphorylated SMAD1, which was giving additional strength to our findings analyzed by qPCR or by immunocytochemistry. And then we were asked to specify the number of mice and their sex in the figure legends for every experiment. So this manuscript has given us the opportunity to continue with future analysis of the role of microRNA 26B regarding ectopic calcification, fibrosis, and sex difference that we are working with right now. And this is the acknowledgement of all the people contributing to this nice manuscript, what I can say, and also the funding agencies contributing, especially acknowledging the British Heart Foundation, and from our side in Navarra, all these entities and these patients association we work with. So I've been said that these these talks are also to promote collaboration. So please reach us out if you are interested on collaborating. As I was saying, we have a huge amount of different samples from tissues, serum, and primary culture cells. And I think that's it. Thank you for your attention. Oh, sorry.
Brilliant talk. Thank you so much.
All right.
That was amazing, Eva. Yeah, that was great. So if you don't mind, Chris, I can kick off the Q&A session. We can see if anyone has questions. And of course, just before we go into the questions, this is also the platform to establish new collaborations. So thank you for reminding us what is available on your side, Eva. And for those that are not aware, we are lucky enough because we have both Eva and Andrea Caporali, which is the senior author on this paper, with us today. So we are open for discussion. If you have any questions, crack on. You can either type them down or unmute yourself while others think. I can maybe ask something myself, if you don't mind, Eva.
Go ahead.
So very, very interesting. I was really, really intrigued by how you found all these phenotypes in smooth muscle cells, but also in fibroblasts and endothelial cells. And in a way, it felt that all of these phenotypes were converging towards this osteogenic state, or promoting calcification that we could probably could link to osteogenic states, which is something we have seen in smooth muscle cells. I was intrigued by, also how endothelial cells and fibroblasts, in a way, and also you kind of showed this again in the end when you were talking about a revision process, how the biological processes showing up are towards osteogenic processes, calcification, matrix regulation, also for endothelial cells. So do you think endothelial cells could directly contribute to this calcification, associated phenotypes?
Yeah, so actually one of the main limitations that we were reporting in the manuscript was that we didn't look at what was happening with endothelial cells or the contribution of inflammatory cells, which was also when I was showing the clustering, the map of the clusters and and the cell types involved in these differences in between knockout and wild type mice. There were some neutrophiles as well, upregulated, and overall T cells. So we didn't include that. Of course, it would be really interesting. And I can't remember from when I was doing my PhD, because this project actually brings me back to what was my PhD, that endothelial cells have been reported to signal in the vascular smooth muscle cell by releasing BMP2. So we could also look at the expression of these endothelial cells as influencers or even senders in this context. But we didn't look at this. It's a thing that we want to work out in the context of valvular calcification, actually. Yeah,
Brilliant.
yeah, it would be really interesting to look at it. Sure.
No, thanks for sharing. And just a link comment to that before I pass on to the others that are coming with great questions. Is the direction I was coming from is your rescue approach in the paper is via LMP. LDN. Well, I got one letter right. And this is kind of working on the BMP pathway and the SMAD1/8 axis. And in a way, if endothelial cells, were involved, I reckon the main pathway being involved would be the canonical TGF-beta SMAD3 pathway. So, in a way, you're not hitting endothelial cell contribution either with your rescue approach. That's why I was curious to know if you were interested into that.
Yeah, but I think it was included at the very end of the presentation as well during the response to the referees, that this LDN has been associated with BMP signaling associated with the signaling of SMAD1/5/8, not the 2/3 signaling. So it would be kind of independent of TGF-beta, as it has been reported previously somewhere else. So we cannot discard, because it could be also cell dependent, but my answer is based on what has been found in the literature previously.
Thank you. So I think I saw Fernando La Cuesta hands. So if you can unmute yourself, and then I will go to Paolo's question. Is that okay?
Yeah.
Let's see if Fernando is still here. Maybe I didn't catch that properly. Paolo, do you want? Yeah, is it you, Fernando?
Yeah, can you hear me now?
Yeah. Please
okay, sorry. Was something wrong with my headphones. So thanks, Eva. It's a very nice piece of work. Thank you. So I was wondering, because it's pretty interesting how your characterization of the model, of the animal, and how the vascular calcification was enhanced by the knockout. But I was wondering, because you focus then you went to see patients with aortic stenosis or aneurysm. So I'm thinking, because I've seen also there's some work in which they have done the double knockout, ApoE and MER26B knockout mice. And it seems you mentioned that there is a link with lipid metabolism, but specifically they were looking at the liver. So I'm thinking, what would happen in the context of atherosclerosis, where the calcification kind of sometimes can be stabilized in the plaque, depending on if it's macro calcification, as you mentioned, and it seems that the females have different phenotypes. So I don't know, what would you expect in a context of atherosclerosis within the aorta?
So, well, as I was saying, the question asked from the referees was very interesting, because it has been found an association of microRNA-26B and lipid metabolism. So as you are saying, because atherosclerosis is associated with lipid accumulation, and so we should expect it's frozen, the screen. I'm not sure you can hear me. Hello?
It's coming through okay? We can hear you okay for now?
Hello.
I'm okay.
Can someone confirm that
I can hear you properly, yes. Can you hear us?
Okay. No, I couldn't hear you. Okay. Thank you. So Well, in this animal model, what we found was mostly associated with medial remodeling, as I was showing by fibrosis and the staining of the histological staining. So it will be more associated to what we found with vascular calcification associated with aging, not with atherosclerosis. But of course, we should have a look at what's happening when you are having a dysregulated lipid metabolism, such as it happens during atherosclerosis, because microRNA 26B has been associated with downregulated lipid profile. But we didn't check that. I expect that it should be associated with, so the downregulation of the microRNA 26B would be associated with an impaired phenotype in terms of calcification. But again, we will be looking at different cell types involved in the process, because this will be affecting more the intima layer instead of the medial one, which is more associated with the calcification of the smooth muscle cell. So I'm not sure I have answered your question.
Yeah,
But...
you did. I think simply, I guess there's a question raised by your work and previous work but it's interesting that it can hit both calcification and lipid metabolism, because maybe it might be worth taking a look what happens in unstable and stable plaques to kind of see if actually it's a marker of instability, which might be very.
Also, because if it's associated with the development of microcalcification, and we know that this microcalcification is associated with higher rates of thrombosis because of the plaque disrupture, etc. We also know from other publications that microRNA 26B has been associated with protective role in the platelet aggregation and activation. So it would be like a very nice combi, let's say, protective combi in the context of cardiovascular disease. So yeah.
Thanks.
Great points. And if I can add on that, because it was such an incredible aspect that I was also thinking about, you know, it looks like very nice the way your data may suggest that the microRNA impairment could be involved in unstable plaques, meaning the microcalcifications are known to be participating in the worsening of atherosclerosis, while wider, bigger, more stable plaque formation seems to stabilize the plaque in a way. So it's nice to see how, you know, when you don't have the microRNA, you are worsening the disease, but it would be nice to maybe see if you had access to like different stages of, which is, you know, is a limitation, different stages of athero.
Yeah, if we can see that this microcalcification progression towards macrocalcification is actually happening, because it's what we found in the human samples. Yeah, that would be nice.
And when the microRNA, you know, is hitting, like, you know, like when it's like the main actor rather than just being a bystander, you know, along the process.
Yeah.
Paolo, do you want to ask the question yourself, or shall I read it?
I'm seeing a few questions.
So I can read the one from
you can read it. You can. First of all, congratulations to Eva, to you for the organization, but just read it and go ahead. It's about my preferred word of adipocytes. Thank you.
No worries. Thank you so much, Paolo. So Paolo is asking whether perivascular adipocytes are expressing the microRNA 26, and if there is any specific adipose adipocyte phenotype in the knockout mice.
We didn't look at that. I don't know if Andrea has been looking lately at this. I don't think so. And I think to remember that when we were doing in situ hybridation, there was an increase of SMAD1 expression in the adipocyte, but I'm not quite sure. I'm talking about like some preliminary tests that we were performing in the tissue characterization in the histological characterization of these samples. So I would say no, we haven't checked. And so I don't know if there is a specific adipocyte phenotype in the knockout mouse.
Thank you. We can discuss it together, because it might be interesting. I'm interested in the perivascular adipose tissue and the effect of the out-in hypothesis, in the sense that, you know, it may affect. We have evidence that it can affect smooth muscle cells migration and other. So can be an additional level of complexity, but very interesting. Yeah. Thank you.
Yeah. Yeah, absolutely. And this could be linked to the obesity and the development of vascular stiffness and so on. Yeah, I mean, the implications could be really, really, really great. But thank you for the comment.
Thank you, Paolo. So I can read the next question from Atito. I hope I'm pronouncing that well. So bear with me because there's a few questions, Eva. They were very interested in your work. So do we also have evidence on the positive role of microRNA 26B when applied in excess, like applying it as a drug, injecting, adding it into a serum? Then they also were asking whether SMAD1, they were also saying SMAD1 was suggested to be regulated by physically cell surface interactions in an endothelial cell specific manner. Having that said, how specific is SMAD1 regulate? How specific is the modulation of SMAD1 by microRNA 26? And are these potentially important alternative pathways that are indirectly also suppressed in the knockout model? Lots of questions. You can start from whichever.
Yeah, so I'm going to start from the beginning. Do we also have evidence on the positive role of microRNA 26B when applied in excess? Upregulation of microRNA 26B was only done when I was explaining all the rationale leading to the development of the hypothesis, a working hypothesis for this project. I was mentioning a previous publication from Andrea's lab in the vasculature. They were upregulating the expression of microRNA 26B in that context, but we did not. We were just targeting the signaling, the BMP ligand receptor signaling. So I don't in our model, I don't know which would be the effect of an excessive overexpression of microRNA 26B. Then injecting, adding into the serum. No, we haven't done either. SMAD1 was suggested to be regulated by physically cell surface contact interactions in an endothelial cell specific manner. Having said that, how specifically is SMAD1? Well, SMAD1 was regulated only in SMAD muscle cells. I was showing that when we were isolating, we were sorting endothelial cells, SMAD muscle cells, and fibroblasts. Only the SMAD muscle cell was upregulating SMAD1 as a result of the knockout of the microRNA 26B. And so we also did some additional experiments where we were silencing the effect of SMAD1 or silencing the expression of microRNA 26B in vitro calcifying SMAD muscle cells to see if there was a mechanistic connection. But I didn't show the data in the presentation, but it is shown in the supplementary material of the manuscript. And are there potentially important alternative pathways that are indirectly suppressed in the knockout model? I don't get this. The potentially important alternative pathways that are indirectly suppressed in the not sure what you mean with this. I'm sorry.
Yeah, I think that they can probably get back to you if they want more details. But my interpretation is that whether additional pathways were modulated other than the one you were directly targeting when you gave for instance, the drug to modulate it. But yeah, that's that's my reading of that question. I'm not sure that's what they want to know.
Well, we actually were asked that from one of the reviewers that was concerned about having regulated a specific regulating the BMP signaling, the different BMP signaling in the by the LDN, but I'm not.
And I think in interest of time, they can ask you because
sorry.
I can see they have a lot of interest. So if they want to get the right information, they can ask you.
Sorry, I couldn't answer that.
No, don't worry. You share a lot of great information with us. I just wanted to give time to others because there are a few more questions. And so Tiana was asking if you comment. So could you comment or mention on options to monitor microRNA 26B expression also in chronic kidney disease and diabetic patients. Do you have any evidence of like knowledge about like the role in those two diseases?
In chronic kidney disease.
And diabetes.
Yeah, I think to remember that there is a previous publication linking the expression of the downregulation of microRNA 26B with kidney fibrosis in the tubular proximal cells. And in diabetic patients, I don't remember right now, but in the kidney, for sure, it was associated with fibrosis. So probably answering to the previous question, I think I couldn't understand for the potential important alternative pathways. I would say that probably fibrosis is also affected by microRNA 26 regulation. And as you know, this might be important in the stiffening, but also in the development of calcification, because at the end of the day, collagen synthesis and fibrosis acts as an early surrogate to the nuclear nucleation of mineral deposits in the vasculature. So I would say fibrosis.
Brilliant. Thank you so much, Eva. Such a beautiful presentation. Congrats to you, Andrea, and the rest of the team,
Thank you.
of course.
Thanks, Andrea.
And
Thank you so much.
Yeah. So I think since we're coming to the end of the session, Chris, we can probably wrap it up. Do you
Yeah.
want to?
Sure. Yeah.
Do the honors?
Thank you again to Eva for taking time to present this amazing work. If anyone,
Thank
of
you for
course,
inviting
has, thank you for being here. If anyone has any other questions, Eva has shared her email here on this last slide. So I'm sure she would love to continue on any kind of discussion with you. Thank you again, and also to Andrea for being present in our talk. And we look forward to seeing you all at the end of October. We'll send you information for the next journal club soon, and please share our flyers again with anyone that you think might be interested.
Yes,
so thank
absolutely.
you all.
Stay tuned and see
Stay
you in a
tuned.
few weeks for an October session. We'll share information soon. Bye, everyone. Thank you for joining.
Bye-bye.
Bye.